Jarrah tree because the bark comes off in long strips, and if you look down on the ground, it's going to make a liar of me. Or look up into the tree, it's got the very small little honky nut. So it might be a prize if someone finds a honky nut. There you go. Sorry, didn't bring a prize for you, don't we? So they're the small ones. That's the Eucalyptus marginata, susceptible species. Not hugely susceptible, but if we do look there, we're actually starting to see a little bit of impact. Is that caused by dieback? Who knows? Could be. Don't know. We're undertaking a bit of an assessment to try and work out whether or not this area is infested with dieback or not. Now, this species over here here we are. So this is Phytophthora umbrosa, the lovely yellow flower. Okay, so this is one of our monocots, so you can't inject this guy because he doesn't have a stem, but you can apply foliar spray to this guy. They are highly susceptible, and if we look just over there on the edge of the path, you can start to see that we've got one there that's just passed away. So we're in our area, we're starting to meet our susceptible species, and we're trying to build a picture as to whether or not this place is infested with dieback. So I'm now going, okay, susceptible species 1, dying. How does the disease get in here? How do we spread it? Vehicles, and exactly right on the track. So you are getting ready for a prize. We have what we call a disease vector, so a pathway into the bush. One of our management areas, we need to start looking at our roads and our tracks. Where are they going? Who's using them? When are they using them? So it's my theory that we've been using this area. We've had potential for, over the last however many decades, disease to be brought in here. It's been spread off the track. Autonomous spread, that root-to-root contact, moving it into the bush. That's my theory, and I'm starting to look around, and I'm starting to say, who else have we got here that might be susceptible? Now these are Xanthorrhoea gracilis, so they're one of the grass trees. This is a small grass tree. It doesn't get a lot bigger than this, and it has the narrow sort of finger thickness. Flower spikes grow to about this big. So these guys are here, but they're healthy, so they're not doing too bad. So I'm starting to wander around. Now I'm cheating and looking at my GPS and looking for ones that I found earlier on. Can't see any, but we will. Just over here, I'm starting to see. You guys can stay there. I'm going to come back in a second. I'm just going to point to it. So here's another one of our Pattersonias, freshly died. Oh, look at this, here he is right here. Here's our little friendly gracilis, just recently died. So he's gone out, and we're remembering that, like our kids growing up, we don't necessarily notice it because it's kind of slow, you know, and they go from being cute and cuddly to stealing your car keys in what seems like a blink of an eye, but it's actually 18 years. And that's a very significant change from being an 18-year-old to being a 18-month-year-old. So we don't necessarily notice it, but the change is significant. So we start to see this sort of thing. Then we turn around again and we see, where's that big, oh, there he is. The freshly dead Jarrah tree just there. And I'm starting to go, okay, I reckon I've got an infested area here. We've got a disease vector that can bring it in. We've got susceptible species in the environment, and those susceptible species are showing disease expression. So they are completely dead. This guy, where's he gone? He's not going to recover. Okay, he's not lying in bed with a bit of lethargy and a snotty nose. He's actually been wiped out. He's not coming back. Okay, he's not like the plants that we can see around that will have, you know, a little bit of death in the crown, perhaps one dead limb, and we will see one down there. So I'm going to say,
I'm just looking at the photograph there that's
and this one here is
just got the top just going. What would you say that is?
looking, it's very difficult. Yeah. So we have lots of things that potentially impact vegetation health. As you know, we have soil nutrients can be variable. We have drought stress that is becoming more and more of an issue. We have potentially insects operating below the surface. Hard to say. I would be suggesting that there's a fair chance this guy is under stress because, yes, I believe he's in an infested area. Okay, so I would suggest, and they do start to yellow and discolour. And when I say like a plant will die like it's had its throat cut, not quite. It's sort of over the course of a couple of weeks, but it's a uniform, consistent, total death across the entire plant in a short time frame. It's not one dead limb, year later another dead limb sort of thing. So I've decided that that tree is in peril, and I'm going to take some straight out. Here we are. So when I undertake treatment, I'm not doing it willy-nilly. I do recognise that I'm potentially impacting a tree. I'm creating a wound. I don't like to go into areas that have no potential risk or no potential threat. I think the actual risk of me doing this is pretty minimal. And if the tree is in the impact zone and potentially going to be impacted by dieback, I reckon it's worthwhile. Oh, unbelievable. Okay, hopefully it'll still work. So that can happen. I've never had it happen before. So on the end of the drill bit, we've got that white cambium. So you can see the white wood. Do you want to run down to the car and in the green bag, the Makita bag, let's just edit this bit out. There's another drill bit, hopefully. Should be in a packet. So, drilling in about 3 to 4 centimetres. The drill is on a slight angle. I'm going to clean the hole out. I'm going to look for a syringe. Okay, I'm going to place the syringe in. We've got about a slight angle going down, just basically to help drainage. It's a spring-loaded syringe, so there's a couple back there you can have a look at. Release the syringe. We don't screw them in, we just place them in. Hopefully it'll make a nice seal. I monitor the hole, because sometimes you don't get a nice clean edge, and it will leak, in which case you need to just readjust. And the tree will take that in over the course of a variable period, which is influenced by the health of the tree, whether the tree is a dominant overstory species or whether it's subdominant and not getting as much sunlight because it's transpiration that is driving the movement. And I don't know if you can see it, but this plunger has already moved about 1 centimetre perhaps in. So the tree is actually actively drinking phosphite, and that is going to go into the stem of the tree, thicken up the stem cells, sorry, not the stem cells, the plant cells, and, sorry? Well, that's how yes, exactly. So it gets in and it's being drawn up and down. Did we? Oh, fantastic. Alright, so we'll just, now we've got that, we'll quickly, so always take a spare drill bit, because we enjoy. And so you'll be able to see, you know, if you've got a dry patch, it'll be coming down one side. Size of the tree, just give him the same number of needles, but squeeze them up. There's no harm in putting another one there if he's got a dry side on the back. You can't be too close. Not a lot of value in putting another one there, because we're just injecting it into the same stream and down to the same part of the roots. Is that a question? Or?
No. Jeremy, would you, because this is actually probably all the same root system, you would inject in all those stems? Those
Yeah.
four?
Yep. Yeah. No, that's right. So this guy is going to take probably 20 needles.
Yep.
You know, when we go and do a contract job, we've got hundreds of needles, and we will go through an area along a disease edge, treating 20 metres, 10 to 15 to 20 metres, depending on the vegetation, either side of that red and green interface that we saw on that disease mapping area, or if we're trying to preserve a specific asset, say, you know, the susceptible species along the driveway of a tourist venture, or if we're trying to protect a rare threatened species of susceptible species in a national park. Parks and Wildlife spray, aerial spray the Stirling Range National Park. We undertake stem injection in areas where there are threatened susceptible communities in parks and wildlife reserves, those sorts of things. So they've deemed an area to be protectable through management, and it's under threat. So
does it go up and down, or does it go both directions? Or just up?
Well, so the xylem and the phloem, I can never remember which one's which, but xylem, anyone know? This could be a prize.
I always remembered it from school, the phlegm comes up.
The phlegm comes up, mate. That means the xylem goes down. But
So it does go, so
the vascular tissue goes both ways. There's transportation of your carbohydrate energy from, you know, your products of photosynthesis, that's going down, and all the moisture and the nutrients are going up.
Yep.
So it's a two-way
So
transportation
that treatment will
system.
go up and down.
Up and down.
Yep.
So you're not bothering to inject marries because they're naturally robust against it.
Yeah, marries are not susceptible. This is not killing the disease. This is our immunisation against the disease, and it's fairly short-lived. So we don't worry about the susceptible species, the unsusceptible species. Just quickly, while we're raising the point of marries, just in case anyone doesn't know, have we got one? Yeah, possibly. But they've got a much chunkier bark as well. So the bark does not pull off in your long linear strips. They've got the big honky nuts, and that's their diagnostic difference. So we'll just wander up here. We're going to have a look at a few more disease indicators, and then we're going to look at the disease edge, take a sample, and then you're free to go. If anyone leaves before that, you'll be in trouble. Different soil types, different rainfall levels, different lengths of daylight hours will result in different growth rates, and that is going to be that trigger of that variability. So you really need to be, undertaking. So if we were to undertake a foliar spray and stem injection program in this area, we'd look at doing. We'd come in, we'd do the foliar spray and the stem injection. We'd do the disease mapping first before anything, work out where we're treating, apply the treatment, and then start looking at managing this area through monitoring over in the next sort of 2 to 3 years. And then when we start seeing disease expression, here's another one of our little disease indicators, the Xanthorrhoea gracilis. So painting that picture again in our mind, that starting to raise the alarm bells, we're in an infested area. We would now start going, okay, are we getting fresh deaths in this area? Have we seen over the last 2 years there are no fresh deaths on our active disease edge? So I want to go and have a look now at a few more disease indicator species, and then we'll go and look at what it's doing in this patch, look at our disease edge, and that's what I say, that's when you're free to go home. So we'll just OH&S, put my safety officer's hat on here. We've got a fresh Xanthorrhea death here, so we're starting to see a little bit more. Podocarpus druinianus or druinianus podocarpus?
Podocarpus druinianus.
You're right. I'm true.
Let's edit that bit out as well. I only want to look highly intelligent. don't want to phone a friend. What was it?
Podocarpus.
Podocarpus druinianus, or as I like to call it, Druinianus podocarpus. This is another one of our disease indicator species, emu bush. Okay, so pretty well. He's not looking too bad. He's looking okay, but we look behind him, and that guy's not looking so crash hot. Pattersonia occidentalis, so the purple flag flower, another highly susceptible species, not looking too bad, you know. So we've got healthy disease indicators in our area, because we're remembering that it doesn't kill everything at the same time. It's impacting vegetation over a period of many years, having a long-term effect. We start moving this way. We've got the Pattersonia umbrosa, so just another indicator, and if we turn around and look over there, we've got the emu bush, or the podocarpus again. So, and that one has just died, literally 5 metres at the base of that. That's a married tree, so we can see the difference in that bark, the married tree as well. So that is starting to be impacted. And now I start to think, what would I be imagining this vegetation type to look like? You know, if there was no disease here, if this was in its natural ecosystem, if this was a naturally unimpacted ecosystem, and I would imagine typically in these parts, on your sort of lateritic sandy soils, I would be anticipating that we would have Jarramari overstory with a Banksia grandis mid-story, perhaps some Persoonia ellipticas, if you know what they are, the snotty gobbles.
Oh, yeah.
And then we're going to have a look at our understory species. So we're meeting our patients, our susceptible species. We've got our Pattersonias, several species of them. We've got our Xanthorrheas. There are several species of them. We've got Xanthorrhea pricii, which is the larger grass tree. There's one just over there. These are the flower spikes of the gracilis. That's how you tell them apart if you're not very good at measuring scale. We've got our emu bush, but I'm not seeing any of the Banksia grandis. So I'm wondering, maybe I've got that wrong perhaps, but we would also consider how long this disease has been here for. If it's spreading autonomously off our disease vector at around about, you know, call it 50 centimetres a year, then it's been here for a significant period of time. We've probably had fires in that period of time. If maybe there were Banksia grandis, maybe I am right, they should be here. Can you see a Banksia grandis? Brilliant. I might have known it was there. But we're going to lose our evidence through fire. That's the other thing. So we now need to start being aware of what's in our bush. So we will now. Oh no, batteries are low. All right, that's cool. So we'll just now start looking, and I don't know if you guys can see it, but we're starting to have pretty much a disease edge. The disease has spread from the disease vector over here through root to root. So it started over there many years ago, and we're going to have quite old deaths over there, and as we get closer and closer to the disease edge, we're going to see the most fresh disease expression. We're going to see the most recent deaths. So now we're looking at what disease indicators are in the area, how they've died all at once, and as a community, we're expecting to see older deaths away from the disease edge and the most recent deaths here. It's like dominoes. Okay, so I got the disease first, I passed it to Mary, Mary passed it to Nathan, yep. Nathan has passed it on and on and on. So I died several years ago, Mary died last year, Nathan died last week. That's that pattern that we see. So we're now looking at individual deaths, the way they've died, radiating out just like a big cup of water might spread down a hill, and through capillary action water might spread very slowly up a hill. So now we look around here and we're seeing very good healthy emu bush, where there are no healthy emu bush over there. We've just got old scaly ones and a full dead one. We're going to head this way and just wander down there, because I can tell you now this is healthy vegetation, this is infested vegetation. We're going to walk down the disease edge so that we don't potentially take infested soil across the border. Okay, so we'll just follow me and we're just going this way. We've got fresh Phytophthora death there. Don't let it distract him, he's on his own. Yes.
What's this level of moisture? Do you think there's much chance of us spreading it?
Okay, so how does it spread?
Well, us picking up soil and moving it from one place to the other.
Have a look at your boots.
Pretty dry, pretty clean.
So,
pretty low chance.
Pretty low chance.
What about orchids, Jeremy?
What about
orchids?
Orchids, as in...
Are they affected by phytophthora?
Oh no. So here's a, just, you know, again, you start to get a bit of an eyeball on what's going on. Here's another Xanthorrhea death. There's a few others over there. But what we're seeing now, if we start looking that way, we're actually seeing what I thought we might see when we came into this vegetation. And that's the mid-story of Banksia grandis, which is just one of the most primarily critical species for so many of our native fauna, as far as a food source, you know, foraging ground for you know, they produce massive amounts of nectar. They produce seed that the cockies like. It's, you know, it's a key environmental species. So we're moving down here. We're looking to our left. We're really seeing a difference between our environments and the de-infested and the uninfested. And
what about peppies?
about what?
How do peppies go with it?
Peppies are unsusceptible to Phytophthora pneumoniae, but they do recover Phytophthora multivora from your peppies. And your tulips as well. See our needles through there at all, anyone? Needles in the tree. Got them? Second one. Make a beeline to them afterwards. So this is your Banksia grandis, and this is your classic sort of disease edge, and this is the sort of pattern of disease infestation that you're looking for. So we've met all of our disease indicator species, being the Pattersonias, the Xantharias, the emu bush or the Podocarpus, the Banksias. There's hundreds of, how many species of Banksia are there, Jane? No points. No points again. Oh. But there's hundreds. There must be hundreds. And now, yeah, now there's Dryanthus. I don't know if anyone knows a guy called Kevin Collins. He runs a Banksia farm, and he has the entire collection of every Banksia in Australia. And about 4 days after he got his final one, they reclassified all the Dryanthus and called them Banksias, and Kev was dark ass. But it's his problem, not ours, so we won't focus on it now. So I'm going to say, look, well, that's where Mary is. Just turn around, Mary, and put your hand on that Banksia grandis there. So this is disease pattern, okay? We don't have any Banksia deaths, grandis deaths down where the car is. We've got one. I'm going to say that guy died 5 or 6 years ago. This guy probably died 2 years ago, and we can see up there we've got healthy living ones. There's one that's got one dead arm on him. Is that a dieback death? No, that's his one dead arm. That's just, he's got COVID, so just stay away from him. He's a dangerous cat, that one. And so this is what we're looking for. So with dieback, we need to be able to be aware of what impact it's having in our bush, so that if we're going to manage it ourselves, we can go in, we can get an understanding of what's happening, what's going on with it. Is it dieback? Is it stress from drought? Is it insects chewing them up and getting in and hammering them that way? Is it something that we can manage? And if it's dieback, we can manage it, and it's going to have this impact. I've got a trained eye, so I don't know if you guys are seeing it, but I'm looking that way and, you know, over there, and I'm just not seeing the volume of healthy plants and everything as I'm seeing that way. It's just not as much biomass in the bush there, and that's our disease edge. So now, when we're talking about management, if I'm going to build a road through here, I'm going to come up to this point, and then I'm going to take a left-hand turn, and I'm going to move my way down through there. So I protect our protectable vegetation. Think of your big cup of water, tip it on the landscape. Water's going to flow from up there, it's going to flow down here. So that area is uninfested and protectable, and we can manage it and keep it that way without any risk to it. If we want to apply a treatment program, we're going to start injecting and doing stem injection foliar spray up in that corner. And James, you get a map that shows you all of this area, all of our evidence, and a disease edge with a spatial data file that you're about to put on your GPS and follow it through. And that will, you know, that would form your disease treatment area that you're then going to look at managing if you're trying to mitigate the spread, stop the spread of the disease. If we're creating a set of mountain bike trails, then really we're trying to avoid going into that protectable area. If we're undertaking the construction of a walk trail, we're going to have a boot cleaning station at this location here, or to be honest, probably 10 metres that way. But we're going to have our hygiene station at a location like this. You inform yourself where this location is by getting your disease mapping done.
Does it travel through sand a lot quicker than clay, say, clay type soil, or vice versa?
Probably, to be honest, in my experience, it travels slower because you've got much more vertical drainage in your sand. Right. So the distribution of water is so in your sandy ecosystems. If you imagine spilling a salt shaker on your table, on your lovely Jarrah table, every grain of salt is phytophthora, and in sandy soils you've got much more sort of clustered pockets of salt, whereas in clay soils it's distributed more evenly across your table surface. And that's sort of how it is. So you get more isolated spots of infestation. I just want to wander across to here. We'll check our needles, see if that guy's taken his medicine. Then I'll show you how to take a sample, because this is kind of tricky. You are reading the bush, you are starting to become familiar with the indicator species, but in a different soil type, in a sand environment or in a clay environment, you're going to have different species. You're going to have the same type of pattern, you're going to have the same disease symptoms, as in straight away dead, but you might want to be able to just confirm what you've got is right. Take a soil and tissue sample, send it to the lab. And again, if you take one of my business cards and you want to find out who the lab is and where they are, just send me an email and I'll send you their details. So,
the Banksy grounders, can you treat them?
Yes,
with
absolutely. So I don't inject anything with a stem smaller than about 100 mils, 10 centimetres. Yeah. The guidelines actually say sort of, you know, more like 140, but that's a fairly decent sized tree. In my thoughts, I give them, I've not killed a tree by stem injecting it. And if it's right on a disease edge and it's a Banksia grandis, it is going to die. So, you know, if we can, yeah. But once they start getting too small, you know, you just don't have the girth, and you are injecting into its cambium and breaking that central flow. And if it is a very small plant, you're creating a much bigger ratio of damage to undamaged. Let's go and have a look at our needles, and we'll take a sample and we'll go on. Side is nearly through, whereas the one near the phone is you know, still got a bit to go. The injection process can take anywhere between, you know, 15 minutes and 4 hours. The eucalypts, they've got I'm guessing now, but they're a much bigger tree, so it just takes longer. They've got slower movement because they're fighting against gravity as well. The Banksias will often take it up within about sort of 10 or 15 minutes, maybe half an hour. So that's your thing. So we're now just sort of wondering, we've got a bit of a picture now. We've come into our area, we're wondering where the dieback's here, we've identified what our susceptible species are, we've seen what we believe is a disease edge. We've got a range of susceptible species, so we've seen disease expression, which is the death of a plant, and we've seen that disease expression through all susceptible species that we've found, but we still don't trust ourselves. We're just sort of making sure that we've got an absolute knowledge of what we've got, so we're going to take a soil and tissue sample and send it off to the lab. Now, being good scientists, we don't want to get any cross-contamination, particularly if we've been taking several samples over several different properties. So I'm using a solution of methylated spirits in here. I use pure methylated spirits in sandy soils, and when you're starting to move into clay soils, the residence time of the methylated spirits in the soil is limited by the evaporative nature of methane. So it's drying off pretty quick. If they're clay, it's not going to penetrate into the middle of soils quick enough to have the sterilising effect. So we add 30% water to our solution. And you can do that with your foot baths as well, just to keep it clean. So this is the similar sort of material. You can use fungicides such as phytoclean, you can use bleach, other things. I don't think, does bleach evaporate? I've never used it.
I don't think so.
No, so that it wouldn't be so good to take a sample because you're going to have a wet axe and it's going to kill whatever you're sampling. So again, using the information we've gleaned, phytophthora in the soil, so there's no value putting the leaf material in. It's in the soil and it's attacked the fine feeder roots, so I don't want to cross-contaminate my sample, so I'm not going to get in there with my boots and start kicking it over and digging a hole with my shoe. So I'm going to use my sterilised axe, just going to move around the edge of the plant. I'm looking for its root system. Each plant has its own specific nature of roots. So these guys have a sort of tufted root system. All the leaves come together at the bottom and create a very hairy mass. Sorry if I'm throwing sand at people. That's a Phytophthora root, so he's got his sort of root ball on him. And we're looking for those fine roots. So we're banging that in the bag. The bag's been labelled with overstory sample 1. I've had issues in the past where just a standard permanent marker was rubbed off in the bag. So I use a paint pen and I just put a plastic label in, so gets to the lab, if they've lost their cover, they can still dig around in Jeremy's bag and find his other label. And I now want to get about half a lunchbox full of soil and root material from around the base of the stem. This is a good example of the guy's root system. I'm going to chuck that whole thing in. Okay, it's soil and root all together.
Jeremy, where do you buy your axe from, your tool?
I think this one got made.
Oh,
They're,
perfect.
yeah, purposely made. But you can just get little mattocks like this. Yeah. I found another one from Bunnings. It's not as good. Yeah. It's invaluable. I use this bit more than that bit. Doesn't need to be sharp. So yeah, this is a custom made jobby.
That's a big sample, man.
It is quite big, but
they want it to be big, don't they, when you send it in?
Yeah, that's really sort of standard. You know, you can get away with less, and if you're working in really gravelly, rocky soils, sometimes you're really struggling to get half that much. But they are culturing a live organism. They're trying to grow it. They're placing it in petri dishes and culturing the pathogen and then applying it to cotyledons of a highly susceptible eucalypt species to actually identify the pathogen under microscopes. So they don't always hit the target. So if you don't give them enough sample material and you want to get the sample rebaited because you're highly confident that it's infested and they send you a result that says negative, which doesn't happen very often because Jeremy never snaps straw bits. But it can happen, and so you want to have enough sample material in there so that if you want them to retest it. So that gets done up with a cable tie which we've left in the car, and I'm now just, can we pass the spray bottle? So we're avoiding cross contamination. I cleaned before and after, just good practice. So that's now sterilised. I've got my my soil point there, taken the sample, fill it in. But what have I done?
Put it all over your boot.
Put it all over my boot. What a clown, eh? You thought this guy'd know what he's talking about. So I'm in an infested area, so I'm going to sterilise the shoes, make sure that when I leave this area I don't have the potential of transporting the disease to another area. I'm just going to borrow your shoulder, my friend. Thank you very much. And this is one method of sterilising. So now I'm effectively pretty happy that I don't have any pathogens living on my boots, and if I walk down this path in dry soil, I'm not going to pick anything else up. This is not a great system if you've got 20 or 30 people, and if you guys are managing, say, winery or a bush area or a local area that you're trying to protect, but you still want people to go in there and be able to enjoy it, then you can apply other tools such as the foot scrubber, which we've got down there, the foot bath, which just has either methylated spirits or bleach or phytoclean in it, and you can have a corral, if you like, a gate that makes everyone go through your one hygiene point, which gives you the clean on entry location. They walk through the foot bath, they walk through the boot scrubber, and they make sure that when they are leaving an area that is uncontrolled and entering an area that is protectable, that they're not transporting the disease in it. So when we leave this area, we're going to go through that gate. We're going to top up that foot bath, make sure that our methylated spirits hasn't evaporated, and we'll all be clean to leave because we've been in an infested area. We made a specific effort not to go into the protectable area. We didn't want to spread the disease in there. Always remember, it spreads in soil, so have a look at your boots. If they're clean, you've performed a hygiene inspection and you're free to proceed. But we just need to be aware of what we're doing and where we're going. That's all from me. Thank you very much for coming along. By raising awareness, we gain understanding, and with that understanding, we can hopefully go out and make a difference. So, good on you. Thanks to Nature Conservation. James, thank you very much for letting us use your place. Thanks to everyone else that did everything that I don't know about. And good on you.
Thanks, Meredith. Yeah, thank you so much.
Cool. And I did forget to say that the Vegetation and Health Service, the VHS, is where you send your samples. And if you want to grab my business card, send me an email, I'll give you all the details.
We will also do a little bit of a follow-up email and send you like the PDF of the protecting your bushland against dieback and a few of the susceptible species, all these other sort of resources. We'll just send it out directly to you as well, which would be cool. But thank you so much, Jeremy. Thank you so much, Meredith, as well. Thank you. You know, very engaging. I know I'm going to take a lot of that back to my own property and also the natural areas that I work as well. It was very good and really nice to see the visual demonstration. So you can read a lot about dieback, but it's not until you're out in the bush and you can hear from it that it's really beneficial. So thank you so much. Excellent.
Pleasure.